Hi everyone. Okay, so this is just going to be kind of a quick overview for chapter 26. It's not a full lecture, but because we ran out of time, I just want to kind of go through and highlight some important things that you should get from this chapter. Most of the chapter is tying in what we've learned with our hormones from the endocrine system, as well as our respiratory system, cardiovascular system, and then our urinary system, which we just recently covered. So there's only a little bit of new information, and I'm going to hit the hot spots that you should definitely make sure that you know for your upcoming exam. So body fluid, when we're talking about that in general, who has the lowest water content and who has the highest water content. So the younger you are, so for example, if you're an infant or a toddler, you would have a higher water concentration in your body, content in your body, relative to somebody who's older. Also, if you're a male, you would have a higher overall water content compared to a female. This is again overall, it's not always the case. Make sure you understand the two main fluid compartments. Basically, it's all of the fluid that's inside of a cell. That's going to be the bulk of the fluid in our body. So this is roughly 2/3 of our overall water content. And then we have extracellular fluid. So any fluid that's outside of the cell is considered extracellular fluid. So there's a bunch of different types. This is only about 1/3 though. Really important is understanding the difference between a non-electrolyte and an electrolyte, because a lot of this chapter is going to talk about osmosis. And if you remember what osmosis is, it's the movement of water from an area of low solute concentration to an area of high solute concentration. So if we're talking about concentration of solids, we could care less what the solute is. What we care about is the number of solutes that we have. So if we're talking about non-electrolytes, these are held together by covalent bonds, and they will not dissociate into individual atoms if we place them into water. So one example of this is going to be glucose. If we take glucose and we put it into water, it's still glucose. We didn't cleave any of the carbon, hydrogen, or oxygen bonds. So we started with one particle, we ended with one particle. We have not changed the particle number, we have not changed the solute concentration, and therefore we have not changed osmolality of that particular fluid. Electrolytes, on the other hand, these are held together by ionic bonds. And when they disassociate, they form ions. That's why we say it's an ionic bond. So if a molecule is held together by ionic bonds, that means the atoms will disassociate when placed into water. So one example would be sodium chloride. We begin with one molecule of sodium chloride, we put it into water, and we end up with two molecules. These molecules are actually ions, so we get a sodium ion and we get a chloride ion. The point is that we began with one particle, sodium chloride, we ended with two particles. Same thing if we take a look at magnesium chloride. It's also held together by an ionic bond. So when we put magnesium chloride into water, it will cleave the ionic bonds and break them. So we disassociate all the atoms. So if we put magnesium chloride into water, we actually end up with three different particles or ions. We get a magnesium ion and two chloride ions. So because of that, magnesium chloride would be considered to have the greatest osmotic power because we are producing more particles when placed in water. So thinking about glucose, sodium chloride, and magnesium chloride and placing them into water, glucose would have the lowest osmotic power, meaning it's not going to cause the movement of water at all. We started with one particle, we ended with one particle. Magnesium chloride would have the highest osmotic power because we started with one particle, we ended with three, meaning that we increase the concentration of solutes with magnesium chloride, which means that we increase the number of particles that we have and increase the osmolarity, which means that water is going to want to move to where magnesium chloride disassociated, again, to try to balance out the concentrations on either side of the plasma membrane. We're usually referring to water moving between the intracellular space and the extracellular space. So make sure you understand the major cation and the major anion for both the extracellular fluid and the intracellular fluid. So our major cation for the extracellular fluid is sodium, and our major cation for the intracellular fluid is going to be potassium. And these concentrations are going to reflect the sodium-potassium pump. So remember, all plasma membranes have the sodium-potassium pump that's going to pump 3 sodium out into the extracellular space for every 2 potassium that it brings in. So this is why we see those high concentrations of sodium outside and the high concentrations of potassium inside. Also make sure that you understand the major anion in the extracellular fluid is chloride, and in the intracellular fluid, the major anion is going to be hydrogen phosphate. So again, the bulk of this chapter is helping tie things together that we learn. If we think about osmolality in the flow of water, if we have a high extracellular fluid osmolality, that means we have a higher concentration of solutes or particles in the extracellular fluid relative to the intracellular fluid. What is this going to do? Well, it's going to cause water to leave the cell and go to the area of higher solute concentration or higher osmolarity, which is the extracellular space or fluid. So water is going to leave the cell and enter into the ECF. Remember, when we're talking about osmosis, we're usually having to move water to make the concentrations of the intracellular fluid and extracellular fluid the same, because the plasma membrane of cells is semi-permeable. So we can't usually change the ion concentrations to balance out the overall concentration. The only thing we can easily move through the plasma membrane is water. So the reverse is true if we're talking about lowering the extracellular fluid osmolality, which would mean that extracellular fluid is really dilute and we have few solute particles. That's going to cause the water to actually leave the extracellular space and go into the cell itself. So usually it's the extracellular fluid concentration that's going to determine the overall intracellular fluid volume and the movement of water. So this is just kind of showing you some things that can cross the membrane. We've already covered all of this. It's just kind of a refresher if you want to take a look at that. Okay, so really important, the amount of water that we have. Usually we need to keep it around 2,500 milliliters per day. So our water intake has to equal our water output. We have several routes at which we can lose waters, including through our skin, through insensible perspiration, and our lungs every time we exhale, as well as in our feces, and the bulk of it, of course, would be in our urine. Water intake is going to come from our food and beverages, obviously, but we also produce a little bit of water with cellular respiration, so we can't forget about that. Remember your cellular respiration equation. We start with a glucose molecule and oxygen. What do we produce? We produce carbon dioxide, we produce ATP, and we produce water. So the water that's produced from cellular respiration via the production of ATP is what we call metabolic water and water of oxidation. You might not think that this water accounts for a lot of overall water that we are bringing into our body, but it actually represents about 10% of our overall water intake, so it's pretty important. Okay, so what they're going to go into next is basically taking everything we've learned now up until this point about osmolality and concentration and applying to what that's going to do to our hormones. So if we have a rise in osmolality, meaning a high concentration of solutes in our extracellular fluid, it's going to cause two things. It's going to activate or stimulate our thirst mechanism, which is controlled by our hypothalamus. And the other thing it's going to do is cause ADH release. So we should know by now what ADH does. It's going to cause aquaporins to be inserted into the distal convoluted tubule in the collecting duct so that we can reabsorb water from our filtrate and put it back into our interstitial fluid and into our blood, which would make sense that we would want to do that if we have too high of a solute concentration or osmolality in our extracellular fluid. As a result, we would produce very concentrated, small amount of urine. If we're talking about a decrease in osmolality, meaning we have a very dilute fluid in our extracellular space, that means we have plenty of water, so we would want to inhibit our thirst mechanism in our hypothalamus and inhibit ADH release, so the excess water stays in our filtrate and eventually leaves our body as urine. So you can go in and take a look at this flow chart that kind of ties everything together if you want. Let's see what else. Again, we've talked about ADH. If we have a low amount of ADH, that means we're going to have dilute urine because we were leaving the water in our filtrate. We aren't reabsorbing hardly any of it, which means we're going to have a drop in the volume of body fluids. And then the reverse would be true. If we have high ADH release, we're going to have a high number of aquaporins inserted at the distal convoluted tubule and collecting duct, which means we're going to reabsorb huge amounts of water, which means the urine that we are going to excrete from our body is going to be heavily concentrated, and we're not going to have a whole lot of volume of it. And again, this is also what it's talking about. You should be very proficient in the RAS system by now. So I will expect you to know how this cascade works, and again, why it's occurring in response to what. Would we have a high osmolality, a low osmolality? And now I would also expect you to know which specific cell and the location of the cell that is going to release renin to cause the cascade into angiotensin II and eventually aldosterone. So there are three principal abnormalities of water balance. One is dehydration, which I'm sure you've heard of. We also have hypotonic hydration and edema. So just a general understanding of what dehydration is. It doesn't necessarily result from not drinking enough water. It could also come from hemorrhaging, because if we're hemorrhaging blood, we're losing blood volume and water. Vomiting, diarrhea, diuretic abuse, and even some endocrine disorders. So if we're dehydrated, kind of understand what's going to happen with water. If we're dehydrated, then that's going to mean that we have very little fluid in our extracellular space. If we have very little fluid in our extracellular space, that means that we're going to have a high concentration of solutes, because the solutes aren't changing, relative to the volume of water, which means that we're going to have a high osmolarity in our extracellular fluid. So how is water going to move? Well, it's going to move from from the inside of the cell out into the extracellular space. What is this going to cause? It's going to cause our cells to shrivel up a little bit. Hypotonic hydration is basically the worst, the reverse of what we just talked about for dehydration. So this is cellular overhydration, and it's actually more dangerous than dehydration. So several different things could cause it. We could start chugging a bunch of water, and our kidneys can't process it fast enough to form our urine. Or if you have any sort of renal insufficiency disorder or disease, that could also contribute to this. Why is this so dangerous? Because we are decreasing the osmolality in our extracellular fluid, because we're lowering the number of particles we have relative to the volume of water. So we have a low concentration. And what this causes is hyponatremia. Hyponatremia doesn't mean we have a low amount of sodium. What it means is that we have a low concentration of sodium. So our sodium content is actually normal, but we've just diluted it so much because we've overhydrated ourselves. And what this can lead to is several metabolic disturbances because the concentration of sodium is not correct. So a lot of times you can treat it with hypertonic saline or something like this. And yes, sometimes it can even cause death. There was like a a game show several times I read where people had to try to win something based off of who could drink the most water, and a couple of the contestants end up passing away because they had hypotonic hydration. This also has happened, you could do a Google search on it, to some marathon runners who drank way too much water along the way and at the end and actually overhydrated themselves. So what happens if we decide to overhydrate ourselves, which is called hypotonic hydration? Our osmolality of our extracellular fluid is going to fall because we're diluting how much solute we have per volume of water. What that's going to cause is it's going to cause water to move to the inside of the cell, which would have a higher solute concentration, and it's going to cause our cells to swell. And they can actually burst if they swell too large. Plants have something to combat this. It's called a cell wall, but we don't have the cell wall, so they can actually lyse our cells if we have too much water entry into them. Okay, edema, the only thing I want to point out here is that this is a disorder of too much water in your interstitial fluid. So it's actually tissue swelling, not cell swelling. Electrolyte balance, really, really important. The most important are probably going to be the ones like sodium, potassium, calcium, and phosphate. Why? Because these salts, which are electrolytes, they control fluid movements, they provide the minerals for excitability. You saw that back in the action potentials for the heart. You saw an AMP1 with your skeletal muscle system, as well as the neuron. And because I'm skipping a lot of this, the only thing that you really need to worry about is the sodium, the hyponatremia, and the hypernatremia. I'm not going to hold you responsible for any of these other electrolyte imbalances, just because we don't have time to cover everything. Okay, going back into sodium, remember it's the most abundant cation that we have in our extracellular fluid. So abundant that out of the total 300 milliosmol of solute concentration we have, it's going to contribute 280 of the 300 milliosmoles. So really, really important. It's really the only cation that's exerting significant osmotic pressure. So if we have fluctuations in sodium, it's going to cause huge movements of water. Our other ions that we have in the extracellular fluid, because they contribute very little to the total osmolality, if we change their concentrations, they're really not going to move too much water. So there is a big difference, and the only thing, again, that we care about really is sodium, between the sodium concentration and the sodium content. So when we're talking about the concentration of sodium, we're really talking about how much sodium do we have per volume of water. And this is going to be dependent on a lot of different things. So if we're talking about overall sodium content, then we're going to be talking about adjusting our different hormone mechanisms that we have for sodium reabsorption or secretion. So if we have alterations in the sodium concentration, what we're going to be trying to change is the volume of water, right? So if we have too low of a sodium concentration, we would need to remove some water from the extracellular space, so that would increase the concentration. If we have too high concentration of sodium in the extracellular space, we would need to add some water to it in order to bring back down that concentration or osmolality. So the primary regulators for sodium concentration are going to be the ones that move water, so ADH and our thirst mechanisms. So we talked about ADH being able to reabsorb water. We would have ADH being released. If we had a high sodium concentration and we needed to dilute it a little bit more, ADH would allow us to reabsorb water to bring that osmolarity down. Thirst mechanisms can also play a role. So we can either inhibit or cause the release of ADH and inhibit or activate our thirst mechanisms in order to control sodium concentration. Overall content we can't really use ADH because the only thing that we can move is water. So if we need to adjust our sodium content, we're going to be looking at the RAS system, which ultimately ends at aldosterone. So aldosterone was responsible for sodium reabsorption from the filtrate to put it back into our interstitial fluid and eventually our blood if we need it. So the RAS system is going to help control sodium content. So if you have a low sodium content in our overall extracellular fluid, we would activate the RAS system so that we can reabsorb more sodium from our filtrate and get it back into our extracellular space where we need it. If we have too much sodium, then we're going to actually activate ANP from our heart. That's going to be released from the atria of our heart so that we can inhibit aldosterone and leave the sodium in our filtrate and get it out of our body. So again, we've already learned this stuff, but kind of you need to tie it together now to what's going on in our body, which system would be activated and which one would not. Okay, so we already kind of talked about this, what would happen. You've learned about this in the hormone system, endocrine system, and you learned it in the cardiovascular system. So if you just need a refresher, you can go through and look at these slides. And same thing here. So these are some other hormones that can regulate sodium balance. Make sure you take a look at these. So for example, estrogen and glucocorticoids, they act to increase sodium reabsorption. So they act very similar to aldosterone. Progesterone helps decrease sodium reabsorption by blocking aldosterone. So progesterone actually acts very similar to the atrial natriuretic peptide, or AMP. So make sure you understand all of the hormones that are involved in sodium reabsorption. And again, the big picture, you can go in and take a look at that. We're skipping over these next couple of slides until we get to acid-base balance. Okay, so picking up at acid-base balance, again, what's really important is that we maintain the pH of all of our body fluids, which usually ranges between 7.35 and 7.45 if we're talking about our blood. Our intracellular fluid is usually neutral at 7.0. But again, what we're talking about and focusing on is our blood. So going back to the pH scale, something that's considered acidic is usually a substance that is lower than 7.0. Is something that's considered basic would be something that has a pH greater than 7.0, and if it is 7.0, then it's going to be neutral. Okay, that's your usual pH scale. But when we're talking about alkalosis and acidosis relative to our body, and in this class and on this exam, we're taking into account the pH that is normally present in our blood. So if we're talking about a normal pH range of 7.35 to 7.45, that is a basic pH. So what we consider acidosis is anything that's below 7.35. So if we had a venous blood pH of 7.3, that would be considered acidosis, even though technically on the pH scale it is basic. Why? Because we're more acidic than we should be in that particular area. Same thing with our arterial blood. If we have a pH of, say, 7.2, what would that indicate? We're still in acidosis because we're more acidic than we should be, even though technically on a pH scale, we're still in the basic range. So acidosis and alkalosis are usually relative to the arterial blood pH and venous blood pH, basically in the range of 7.35 to 7.45. If we fall outside of that range, we are either going to be in acidosis or alkalosis, and we're going to need our buffers to come into play, again, to help maintain that very narrow pH range. I also want to point out that the venous blood is a little bit more acidic at 7.35 relative to our arterial blood. Again, arterial blood is the blood that we're bringing from our heart that's heavily oxygenated. It's going to go into our capillaries of our tissues where it's going to unload the oxygen and pick up waste products. and it's going to exit the capillary bed in the tissue via your veins. So the venous blood has a slightly more acidic pH because of the higher acidic metabolites being produced as a result of metabolism. So things like carbon dioxide, which is eventually going to produce carbonic acid within the cells, plus some other uric acid and things that are produced as waste products. Those are acidic waste products being produced that are being picked up at the venous end. Okay, so we have three buffer systems that are important. Chemical buffer systems, we have them readily available, ready to act. We've already talked about one, that was the carbonic acid bicarbonate buffer system. We'll talk about two more chemical buffer systems we have. Our brain can also act in helping with our fluid pH by either increasing or decreasing our respiration rate. Carbon dioxide does act as an acid because it forms carbonic acid. So we can exhale and change our ventilation rate more carbon dioxide in order to bring that back down to a normal pH range. The downside to using our respiratory rate to adjust for pH is that the only thing that we can actually get rid of and alter are carbon dioxide. We can't actually get rid of any of the other metabolic waste products like uric acid via our respiration. So we still have to figure out something else to get rid of all of those other acidic waste products that we have. And that's where our kidneys are going to come into play. So our renal mechanism is going to be the most potent. Downside is it takes a while for us to adjust for the sensors to get the information to our kidneys to adjust what we're putting into our filtrate and what we're reabsorbing. So chemical buffer systems, again, a buffer in general is something that can act as an acid or act as a base. So what their goal is, if we take something that's really acidic, we're not really getting rid of it. We're taking something that's heavily acidic, a strong acid, and the buffer is going to convert it into a weak acid. If we kept the strong acid, we would have huge fluctuations in our pH of that fluid. But if we can turn that strong acid into a weak acid, then we're going to have a very small fluctuation in our pH. And the same thing when it acts as a base. It can take strong bases and basically turn them into a weak base so that we have a very small pH change instead of a very large pH change. So what's the difference between a strong base and a strong acid and a weak base and a weak acid? Well, it's its ability to dissociate in water. So here's hydrochloric acid up here. Hydrochloric acid is considered a strong acid because when it's placed into water, it completely disassociates into its ions. So you can see we have no hydrochloric acid left. When we put it in water, all we get are hydrogen ions and chloride ions. A weak acid, like carbonic acid, which we've talked about before, it's a weak acid because it only partially disassociates. So you see we only generated 2 hydrogen ions and 2 bicarbonate ions here. Some of it remains intact. So if you remember what the pH scale is, the pH scale is the negative logarithm of the hydrogen ion concentration. The higher the hydrogen ion concentration, the lower the pH and the more acidic something is. So if we look at the hydrochloric acid when it's in water, we produce a high number of hydrogen ions, which means this is a very strong acid and would have a very low pH because it has a high hydrogen ion concentration. If we're talking about a weak acid like carbonic acid, when it disassociates, we only produced 2 of these hydrogen ions, so a very low hydrogen ion concentration, which would indicate that it would have a little bit higher of a pH than our hydrochloric acid. And we could use it to an advantage by, again, not causing as much of a swing in our overall fluid pH. So bases act basically the same way. So chemical buffer systems in our body, we have 3 major ones. We have bicarbonate, carbonic acid, buffer system, you can go in and look at this. I do expect you to know every single thing about the carbonic acid bicarbonate buffer system just because we've worked with it so much. This should be a refresher for you, but if it's not, make sure you take a look at this and understand what's going on. The only thing I'm going to point out right here is that we start with a strong acid. When we use this buffer system, we turn the strong acid into a weak acid. So instead of going through a major pH change with the strong acid in the fluid, now we're going to go through a very minor pH change because we've converted that strong acid into a weak acid. Same thing now, they're telling you basically we turn a strong base using the bicarbonate buffer system into a weak base, again, to only cause a very minor fluctuation and a change in pH. The other one that we have is the phosphate buffer system, which acts just like our bicarbonate ion buffer system. The only real difference is what the salts are, what the acid and base are. So the sodium salt is going to be the dihydrogen phosphate, so the one with 2 phosphates, and monohydrogen phosphate that acts as a weak acid. A weak base. But the overall point is the same. We can use dihydrogen phosphate and monohydrogen phosphate as a buffer. It can act as both an acid and a base. And what it can do is it can convert a strong acid into a weak acid, and it can turn a strong base into a weak base so that we only cause minor fluctuations in the pH itself. The third type of chemical buffer system that we have is actually using our proteins. So we consider proteins an amphoteric molecule. An amphoteric molecule means that it has 2 regions that can do something different. So one portion or one segment of a protein can act as a weak acid. The other segment or portion of this molecule can act as a weak base. So I drew for you previously the structure of an amino acid, which is basically carbon, a hydrogen, and then an R group, this is dependent on what amino acid it is. But what's always the same on every amino acid is that it has an amine group and it has a carboxylic acid. So that's why we call it an amino for the amine group acid. So the amine group can actually function as a weak base. So we can tie up an additional hydrogen ion to form ammonia. And then our carboxylic acid can act as an acid and donate its hydrogen ion to increase the pH of something. So proteins also, because they're amphoteric molecules, can act as a buffer. So again, make sure you know everything about the carbonic acid bicarbonate buffer system. Know that phosphate can also be used. Monohydrogen and dihydrogen phosphate can also be used as a buffer. And even our proteins can be used as a buffer using its amine group and its carboxylic acid group. The purpose of the buffers are to turn strong acids and bases into weak acids and bases so that we can only have small fluctuations in pH changes. Okay, so this is a really good kind of overview slide. I also meant to point out you should understand where these buffer systems are primarily working. So this is our main extracellular fluid buffer, the carbonic acid bicarbonate buffer system. Remember, we make the bicarbonate ion inside cells, but then we shuttle it outside immediately. If we're talking about the phosphate buffer system, why would we use this one versus bicarbonate? Because we find a lot of these products in our urine as well as our intracellular fluid. In proteins, this is going to be the most important one in the intracellular fluid, as well as the blood plasma, because this is where the bulk of the proteins are going to be located. So like I mentioned, kind of what you should get from the respiratory regulation is we can alter our respiratory rate based off of the acidity that we have in our blood. So we can either cause us to retain more carbon dioxide to make our blood more acidic if it's too basic, or it can release more carbon dioxide in order to make our blood more basic in the event that it becomes too acidic. The only thing downside to this is it does act pretty quickly. But again, like I mentioned, the downside is the only thing that we can alter is carbon dioxide. So if we have acidosis as a result of another molecule, say uric acid, altering our respiration rate isn't going to do us any good because we can't eliminate the uric acid. So the only thing that we can change is really the concentration of carbon dioxide that we have. So you can go in and take a look at that. I'm not going to go. Okay, the renal system and the urinary system, this is going to be, again, our major one. Let me go back up here. So the renal regulation is going to be our ultimate acid-base regulatory organ. Like I said, the downside is that the kidneys act pretty slowly. So the chemical buffers cannot eliminate acids or bases from the body. Their only function of the bicarbonate ion, phosphate, and protein buffer systems are really just to temporarily adjust our pH, right? They're not actually eliminating the excess hydrogen atoms, and they're not really consistently producing hydrogen atoms to do it. It's only a temporary fix. So we really need our kidneys to either go in and reabsorb things or keep things in our filtrate to maintain our overall blood pH. So like I mentioned, we can use our lungs to help eliminate carbon dioxide. Downside is we can't get rid of other acids that are generated by cellular metabolism. So we need our kidneys to come in to actually eliminate the phosphoric and uric and lactic acids that have been produced in order for us to prevent continual acidosis. So when we're talking about excreting bicarbonate ion, every time we excrete a bicarbonate ion, it's the same thing as gaining one hydrogen ion. And I'll show you something that will hopefully make it less confusing. So in order to reabsorb bicarbonate, the kidney must secrete hydrogen. And to excrete excess bicarbonate, the kidney actually has to retain a hydrogen atom. So we only lose one, and then we only keep one of them. We don't keep both, and we don't lose both. And in that manner, we can adjust and alter and maintain our pH of our blood. So again, we've already talked about the bicarbonate buffer system, but we'll just keep it going here. Make sure you take a look at this. So what this is showing you, if we start at 1, we have carbon dioxide combining with water in our cell like we've seen 1,000 times before. We have carbonic anhydrase that's going to convert these into carbonic acid. Carbonic acid is incredibly unstable and is going to dissociate into the bicarbonate ion and a hydrogen ion. The bicarbonate ion is going to be pumped out to go to our blood to act as a buffer. The hydrogen ion is going to get pumped out to our filtrate. When it's in our filtrate, it can combine with a bicarbonate ion there, reforming carbonic acid and splitting into water and carbon dioxide. The benefit of this is that the carbon dioxide can then enter back into the cell to complete this process all over again. So we've already talked about this. Now we're talking about it in reference to the filtrate relative to the erythrocyte and other things that we've talked about as well. So that's really great that we can recycle most of the bicarbonate that we have through the process that I showed you, but sometimes we don't have enough bicarbonate. Say, for example, we get into severe acidosis or something else happens with our cells that are capable of producing bicarbonate ion. So we have to have mechanisms in order to generate new bicarbonate ions that aren't necessarily coming from carbon dioxide. So we have 2 different mechanisms. The first one's going to be via excretion of buffered pH, and the other one's going to be from ammonium excretion. So again, just have a general understanding of what these are. I just kind of want to point out that there's 2 mechanisms. These are occurring in the proximal convoluted tubule. And I mentioned that there's 2 types of intercalated cells in the collecting duct, type A and type B. I told you just to hold that thought right now. We'll talk about them again. So the type A intercalated cells in the collecting duct, these are capable of generating new bicarbonate by getting rid of hydrogen ions. So here's what it's showing you here. We have water and carbon dioxide that's forming. Right here, we have carbonic anhydrase that's coming in. Then we are going to form carbonic acid, which is going to split our bicarbonate ions, still going to shift and go into the blood. And if we look at the hydrogen ion, it's going to go out into our filtrate and combine with hydrogen phosphate to form dihydrogen phosphate that's eventually going to be eliminated into our body. So if you notice, we're taking carbon dioxide in this case that's being generated from cellular metabolism in order to produce the bicarbonate ion. Previously, when we looked up here, the carbon dioxide that we were producing the bicarbonate ion from was coming from our filtrate. So that's the difference, and this is how we can generate new ones, is not by recycling, but taking the carbon dioxide from cellular respiration and turning it into a brand new bicarbonate ion to get it into our bloodstream. The other mechanism we have is by using an amino acid. We can use glutamine. So if we have glutamine going into our filtrate, we can reabsorb glutamine into our proximal convoluted tubule cell, where it's going to undergo a series of reactions like deamination, oxidation, and acidification, and then it's going to split. And when it splits, it's going to produce 2 ammonium ions, which are just going to be secreted out into the filtrate and eventually leave our body. But the good thing about using glutamine is not only do we produce 2 ammonium ions, we produce 2 bicarbonate ions. So it's a little bit more powerful in the production of new bicarbonate ions. Again, don't get caught up in all the crazy details. Just understand the mechanisms that we produce new bicarbonate ions. One is from using carbon dioxide that's being produced from cellular metabolism within the proximal convoluted tubule cell itself and generating bicarbonate that's going to enter our blood. We're not recycling it and picking it up from the filtrate. The other method that we can use is we can take an amino acid, glutamine specifically, reabsorb it, go through a series of reactions in which we produce 2 bicarbonate ions along with 2 ammonium ions, which are just going to be secreted into our filtrate. So all of that was occurring when we're talking about producing new bicarbonate ions in our type A intercalated cells. But if our body is in alkalosis, meaning it's too basic, that means we need to get rid of some of that bicarbonate ion in our filtrate. So our other intercalated cell, intercalated type B cell, is going to be responsible for secreting bicarbonate ion from our filtrate, secreting it from our blood and into our filtrate so that we can go in and increase our blood pH. Every time we secrete a bicarbonate ion, we are going to reclaim a hydrogen ion, because if we lose one, we gain one. So also reclaiming the hydrogen ion is going to help us increase the acidity or lower the pH of our blood, which is the basic goal. So basically it's a flip type A cell. Okay, I think that's it. Is there anything else I wanted to mention? Okay, that's it. Hopefully that's helpful. When I see you on Thursday, if you have any questions, let me know.